It's 20 minutes, right?
Yeah, 20, 25.
Okay. Well, this is a talk I gave last week, week before at AFS, American Fisheries Society. And I'm giving you some, the first sort of real results from Atlantis modeling of the Deepwater Horizon oil spill that we're doing with the Sea-Image and Sea-Image 2 project. Also with a little bit of cooperation from the Deep Sea Consortium. So I guess a lot of you probably haven't heard of Atlantis, so apologies to those who've heard me talk about it before, but it's a fisheries and food web model. It's spatially explicit in 3 dimensions. It uses an irregular polygon grid. The species in the ecosystem are summarized into functional groups, which are groups of species aggregated by trophic and niche similarity. It's fully age-structured for vertebrate groups. It includes a gape-limited feeding algorithm and explicit handling of the detritus and nutrient dynamics. Also contains a link to hydrodynamics model, so the lower trophic levels are affected by physical ocean data. We just completed a NOAA technical memorandum earlier this year in cooperation with the Southeast Center. It summarized the data that went into the model, as well as some exploratory simulations and diagnostic tests. So I want to highlight a few of the methodological papers that we've published over the last few years. The first was funded by Florida Sea Grant, and it was a food web study. We sampled about 1,200 stomachs. We also collated data from FishBase, Sea Life Base, and the FWRI database. So we had a wide variety of stomach content information, and we applied a statistical method that I developed in 2011, which uses fitting using a model called a Dirichlet, which is a multinomial beta model. The beta distribution is bounded at 0 and 1, so it's often used for proportional diet data, and it's flexible and can take a variety of shapes. The reason we use the Dirichlet is because it accounts for covariation between prey items. So the statistical fits provided so this would be for one predator, and each of these colors is one of the prey items that it eats. The statistical fits provide, which we use a maximum likelihood estimation procedure, provides the modes, which are the contribution of the prey items. So that would all add up to 1 for any predator. So my student Michelle Massey published this in 2014 in Ecological Modeling. This was one of the outputs of that. It's a food web for the Gulf of Mexico as a whole, which we then used we used in the Atlantis model. Subsequent to that, we had a second diet project, which was funded by Marfin, and this is ongoing, but we've already produced our main deliverables. So we've included an additional 1,000 stomachs. We funded FWC to do this diet sampling for us, and we directed them to sample the species that we had that represent data gaps in the Atlantis model. The outputs from this study have been now integrated into the FWC database, as well as the GOM Trophic Interactions database, which is a food web database hosted at Texas A&M Corpus Christi. Then for fun, so we improved the diet study, we improved the diet matrix, and just for fun I dropped it into the Atlantis model. I didn't necessarily expect an improvement in dynamics because it takes quite a bit of tuning to get the full advantage of the more accurate diet matrix, but to my surprise it improved the dynamics quite a bit just right out of the box. So this bar chart here shows the improvement in residuals in a historical simulation from 1980 to 2010 relative to observed biomass and catch. So the red bars show where we were before, the blue bars show the residuals after we've integrated the new data. And about 2/3 of the groups improved. There's a 23% reduction in the residuals and a 28% reduction in bias. So that was a pleasant surprise. That diet matrix is now integrated and included in all the results that you'll see today. So those two studies formed our diet matrix. Then we also came up with a statistical procedure to produce biomass distributions for Atlantis. This was work that was led by Mike Drexler, my PhD student. We developed a generalized additive model, or a GAM, which predicted the biomass distributions based on these 5 physical properties: bottom temperature, oxygen, depth, sediment type, and chlorophyll A. We used the GAM to estimate the abundance densities at 1/10 degree resolution, and then these were averaged up to the level of our Atlantis polygons and used to initialize the model. So that GAM has actually been used in a number of other applications. This is one of them. The second paper, which is was just completed by Mike Drexler, it provides connectivity for the larval dispersal matrix in Atlantis. So he developed, based on some earlier work I did, an individual-based model of larval transport, which uses hydrodynamics, currents from a hydrodynamics model to move particles around. We're integrating the currents over, I think, the first 50 meters or something, so it's mainly based on surface currents. But considering that so we're missing larval vertical migration behavior, and we acknowledge that, but we are doing it for about 47 species, so there just isn't the data there. For a small number of species, I think 5 species, we are using better data, which were provided to us by NOAA, but for the rest we're using this simple IBM. So the particles move around, and the weight of the particles is proportional to the adult spawning biomass at the source, which we take from that GAM model. So from that, we get connectivity maps, we get larval tracks, and we are able to produce a source sink matrix, which we can use in Atlantis. And what that does is it apportions the recruitment rather than having the babies recruit at the adult locations. They recruit at more realistic settlement grounds. This is some of the diagnostic tests that are included in the tech memo. We produced equilibrium catch and biomass curves up here, which are equivalent to the type you would see in a single species stock assessment. So from this we can estimate the MSY and the B0, or the virgin biomass of these species. And that's a way of confirming that the productivity of these groups falls within a realistic range and the behavior is comparable to a single species model. In addition, we did historical reconstructions from 1980 to 2010. I'm just showing you, I've cut off the first 10 years here, as there's a little bit of transient dynamics that has to work itself out. But overall, the fits to the historical data were satisfactory. So now moving on, so that's development of Atlantis, and there's basically 2 projects right now that we're using Atlantis for. One, which I'm not presenting on today, is linked with the Integrated Ecosystem Assessment, the IEA project that NOAA has, which is basically sort of their formal umbrella for ecosystem-based management. And that's work that Michelle Massey is doing and actually is producing some very interesting results, which I'll have to present another day. So what I'm showing you today are results from our oil study. So we have modeled the Deepwater Horizon oil spill as we think it happened. The effects we've included, so first our fish toxicology effects, which includes growth, mortality, and recruitment. We've also included a zooplankton bloom. Kendra tells me that there's evidence of a zooplankton bloom in the latter half of 2010. And so what I've done here is I've reduced the natural mortality term for a suite of zooplankton groups. And to show you what the effect is, this is what's happening here in the large zooplankton. So I've calibrated it to represent a 10% increase in the zooplankton density immediately following the deepwater horizon. And this is just a reaction to the abundance of planktivores, as you'll see in a minute. The other groups, we applied this to small zooplankton and some others, but the spike in large zooplankton actually suppresses the small zooplankton because of a trophic effect. So this is the key effect that we're representing. And finally, fisheries closures. After the spill, there was an immediate closure in a box surrounding the oiled area, and in addition to that, some loss of consumer confidence resulted in reduced fishing effort, so that is in the model as well. Effects not included. What you're seeing here is quite preliminary. We haven't included invertebrate toxicology. It's only the effect we've only applied to fish. There's also very likely some benthic toxicology and resuspension effects that we haven't attempted to model, nor oxygen limitation. The current understanding is that oiled marine snow fell out at a very quick rate and accumulated on the bottom, potentially leading to reduced oxygen levels in the sediment, which could ultimately affect the demersal food web. So we haven't really modeled that. We're just modeling water column effects, and so that's a caveat that I'll remind you of later when we see some results. So first, I'll talk about the recruitment effects. We've taken this directly from work by Emily Chancellor, who's one of Steve Murawski's students. She's used surface observations of oil and overlaid that against ichthyoplankton estimates from CMAP to come up with a relative index by taxa of the impacts on recruitment. And so we have, you see this data here in the box and whisker plot showing the species most seriously impacted. So we've aggregated this into Atlantis functional groups and are using that as a relative index of recruitment impairment. The effects on growth and mortality were developed by Lindsay Dornberger, who's my PhD student. She presented this paper at Gomery in Houston in January, and Houston? Yeah, it's Houston, right? Sorry, it's a few trips and they're blending together. And she got awarded the best student paper along with Christina Deek. And so we have used an AIC model selection procedure to develop functional responses. And in fact, for mortality, the one that won was a hockey stick formulation, which includes a threshold below which there's no effects and then a linear increase. New work just completed last week actually indicates that for the growth effects, the exponential curve is slightly better, but for the results you'll see today, we've used a hockey stick for that one as well. So those models are fit to our data from literature. Right now, at the time we did this, no results from CImage that were usable were available. So this is based largely on data from Alaska and some laboratory experiments and one other oil spill, which I can't remember right now. So this is not from, these oil effects are not from the deepwater horizon, but they should suit our purpose. Lindsay is going to take a second stab at this and also resolve the effects into reef, pelagic, and demersal components, whereas this is just using a single relationship for all fish species. So once we have the functional response, we can use as our independent variable oil concentrations, which we're getting from Claire Paris. She has a Lagrangian particle transport model, which she's using for oil fate. Claire Paris is a professor at the University of Miami. And her modeling system also includes microbial degradation. So what we see here on the right are just a few screen captures from her simulations. And this is all done explicitly at depth. So we have oil concentrations at various depths, and Atlantis is also explicit in the vertical dimension. All right, a few caveats. So first of all, the oil data that we had tend to be from chronically exposed areas, so there is a possibility that some of those organisms have resistance. The data tend to relate to sediment concentrations, and all we had was the water column. So what we've done are we've integrated the oil density through the water column and assume that's representative of the sediment concentrations. So those are the two main caveats to this work. And I think eventually we'd like to use a uptake deparation model, but we don't have that in place. I want Lindsay to do that for her thesis. So right now we're just using the instantaneous effect from the ambient oil concentration. And because of those, what we're about to see are conservative estimates, I think. Sea image 2 has got some very fancy, well thought out ecotoxicology experiments, which should really give us a much better estimate of how the oil impacts the biota. So this is a result showing groups of groups. So Atlantis has 91 functional groups, and we've aggregated some of these to just give you an idea of what's happening. We see about sometimes as much as a 1 or 1.5% decrease in overall biomass for that group, but as I said, these are groups of groups, so some would be more or less, some of the constituent groups would be more or less than that. The most affected groups we see are elasmobranchs, gag, red snapper, Lutjanidae, which is other Lutjanidae, and other demersals. So what we have here is our 100-day oil exposure, which is this shaded area, and then we turn off the oil effect, and the rest of it is all trophic interactions. Now you can see for shrimp that they go down and then go up, but recall that I don't have any benthic, I don't have any effects on invertebrates, and I don't have the resuspension effect. So it's very likely that they're going down, and that's just from the effect of the fish. So we'll expect to see some decreases.
Can you explain the ordinate to us?
This is relative to a no oil scenario. So this is with...
We're looking at 0.5%. So what are the error bars on that?
Well, yeah, so as I said, these are groups of groups. So like contained in the elasmobranch are about like 6 or 7 different functional groups. I didn't put the error bars on that, but on the next slide we'll see.
My point is that, let's see, it goes down to, elasmobranchs go down to 98.4 or 3. Are you telling me the error bars in this model are such that you can, that a 1.7% reduction is significant?
Well, as you'll see, it's hard. I'm not sure how to respond to that. As you'll see, the recovery times are significant. We are now in the process of some sensitivity analysis to make sure. So this, I think, is sort of the best case scenario. All I can say is that we're working on sensitivity analysis. So these are individual species. So you can classify the results in a few different patterns. This is what I call direct recovery here. So there's an initial decrease. This happens in about 25% of our groups. An initial decrease and then a direct recovery to the pre-spill condition. And then in a surprisingly large fraction of the groups, about 40% of the groups, we see the initial decrease, we see the recovery, but it never quite makes it back up and instead takes a downward dip. And so I interpret this to be good evidence of trophic cascades, as it does happen in 40% of our groups. The reciprocal cascade can be, and this tends to be in predator groups, as you can see here, tuna, ceranids, lucanids, and here's red grouper. There is a reciprocal cascade in some of the smaller prey species, menhaden, this is a demersal fish group, and this is a small reef fish group. So I think this is just responding to the fluctuations in these piscivores, and then there are some species that aren't directly impacted by the spill. Now, again, I don't have invertebrate effects, but this is this one and this one are all shrimp groups. So we see an increase initially, which is the result of the loss of fish, followed by a decrease. In about 8% of the groups, we see something concerning. There is a decrease followed by an increase, but it never comes back to the level that we had before the spill, indicating potentially an alternative stable state. So if we look at this as a whole, these recovery times as a whole, we see a bimodal distribution of recovery times. So these would be the direct recovery species. These would be the species that are engaged in trophic cascades, and they take a little while to work out those problems. So of the direct recovery, we have about 25% of our groups. The species that are active in these trophic cascades, about 40% of our groups recovering within 25 years. About 28% of the groups don't go down at all, and about 8% of the groups don't recover to their pre-spill levels. The assumption that we made concerning the large zooplankton turns out to be rather important. So we've increased here the abundance of large zooplankton initially. This actually results in a decrease in the small zooplankton, and because of that, planktivores decrease, and we see a reduction in the piscivore to planktivore ratio. And an increase in the mean trophic level of the system. One other thing, so here we're showing condition factor. There seems to be a reduced condition factor for pelagic fish, indicating that they're having a more difficult time feeding. We don't see that effect in demersal fish, but that result could change once we include the effect of marine snow, of oiled marine snow, and resuspension. I heard recently that about 14% of the oil is thought to be still on the bottom of the Gulf.